Hello, everyone. Let's continue with our next webinar, presented by Bingrong Chen. Regulatory considerations in raw material selection for cell and gene therapy manufacturing. Click the speaker information tab on the left of your screen for a complete biography on our speaker. Again, we encourage you to submit questions during the presentation, and we will answer as many as we have time for afterward. Bingrong, you may begin your presentation.
Thank you so much, Casey, for the introduction. And it's actually a pleasure to be here with the team as well to present on my presentation on regulatory considerations in raw material selection for cell and gene therapy manufacturing. So once again, I'm Bingrong here, the Regulatory Affairs Manager for Thermo Fisher. Okay, so in the following presentation, I'll be actually covering the following contents. So I'll be starting off with the terminologies and guidance, the regulatory considerations for ancillary materials, ISO 20399 and USP 1043 standards, the cell therapy products that we have at Thermo Fisher, as well as ending off with the regulatory document support for clinical and commercialization manufacturing, and the conclusion. So for ancillary materials, also called as raw materials in Europe, there are components that are used in manufacturing but not intended to be present in the final product formulation. For consistency in its sharing, I'll refer to the term ancillary materials. USB 1043 and EP 5.2.12 are reference guidance for ancillary materials. Example of such includes the cell culture media, cytokines, serum, and buffers. Although ancillary materials are not supposed to be present in the final CGT product, such materials can affect expansion, differentiation, and activity of a cell and tissue product, which in turn can have significant impact on the final product quality attributes. Therefore, it is important for CGT manufacturers to carefully define the selection criteria and subsequent qualification of ancillary materials. So the main guidance around ancillary materials are USP 1043, EP 5.2.12, and ISO 20399. They provide the process of acquiring and evaluating data to establish the source, identity, purity, biological safety, and overall suitability of a specific ancillary material for use. USP 1043 also provides guidance for developing appropriate ancillary material qualification programs. It's to note that ancillary materials are not regulated on its own, and they are assessed as part of the final CGT product. USP 1046 provides information on good practices to overcome such challenges in manufacturing, characterization, and testing of the cell therapy products. The chapter also discusses potential impacts of ancillary materials on final product quality. Other general chapters in USP provides details about conducting tests associated with the different types of ancillary materials. Example of such includes the USP 90, 92, and 1024. Test specifications and validated test methods are also provided in the pharmacopoeia. The criticality of ancillary materials depends on the application for which the ancillary material is used. So testing of the ancillary material against a pharmacopoeia standard ensures conformity to the identity and functionality. Regulatory guidance and standards issued by international regulatory bodies such as ICH and ISO serves as starting points for evaluating, selecting, and qualifying such ancillary materials. So similarly, these are the same standards that Thermo Fisher use as part of our development and testing of ancillary materials. In terms of regulatory guidance, this year, the US FDA issued 3 main guidance, providing recommendations and information on gene editing, early phase clinical trial and CAR-T therapies, potency assurance for CGT products. So such guidance highlights recommendations in CMC, pharmacology and toxicology, clinical study designs to enable fast registration and launch of such products. The WHO also rolled out a CGT guidance in an attempt to harmonize the regulatory guidances. So for early phase 1 to 2 clinical trials, especially in first-in-human clinical trials, the objective is always an evaluation in terms of safety, the nature of potential adverse reactions, and estimated reactions to dose and pharmacological reactions. This includes gathering data and clinical evaluation to support future commercialization. There might be some features of a phase 2 clinical design in the preliminary evidence of efficacy, but the main assessment is always on the trial risk and safety evaluation. As CGT manufacturers process to late phase trials, there will be a greater emphasis on CMC data and characterization. So this will include the raw material characterization, in-process testing, and batch release specification of the final products. Beyond phase 1 clinical trial, the manufacturer also needs to demonstrate the ability to remove these ancillary materials from their final products. So the manufacturer should have a thorough understanding on the quality of media and raw materials used for cell and gene therapy because of the challenges that's used to perform the end-stage sterilization. The use of IUO ancillary materials require more qualification to ensure that the material is suitable for its intended use. This validation and qualification of IUO materials can lead to significant additional cost. So it's advisable to use GMP-compliant material from the start to ensure that the end product is compliant to the local regulation. The entry point to make the switch from IUO to ancillary materials depends on the risk and cost sensitivity. The switch from IUO to GMP materials such as CTS branded products, which I'll be covering in minutes' time, should be considered as soon as a target has been identified. So in US, most of the investigational products in the phase 1 clinical trial are exempted from complying to CGMP standards, but in other countries such as EU, Singapore, for example, the use of CGT ancillary materials will be required from the start of clinical trial. So by choosing such reagents or components that's able to meet the rigorous regulatory requirements, we avoid the need for additional changes that will need to come later, and this will actually reduce the amount of time as well as cost. So changes in methods or switching of a critical ancillary materials from different phases of the clinical trial will require very lengthy and costly comparability studies per ICH Q5E. The TAP Center for Study of Drug Development estimated that the cost to an amendment of a phase 3 clinical trial will cost 3 times as much as an amendment of a phase 2 clinical trial. So clinical changes such as the process changes or composition of ancillary materials during this clinical development will directly impact the quality of CGT products. So this can impact the validity of previous studies undertaken before the change. So changes made once the product has entered into the clinical phase should also be assessed for their impact on regulatory compliance, and for the critical changes, such approvals will be required from the authority before the implementation of the change. The data required to justify for such changes would be proportional to the stage of development and the level of risk. It is therefore important to establish a very close relationship with your suppliers at early stage and to optimize such products and protocol as necessary. So although ancillary materials are not regulated on their own, regulators are still required to demonstrate the ancillary material qualification. The specific ancillary material qualification process depends on many factors, including the type of ancillary material, the type of cell therapy products being manufactured, and the stage of manufacture in which the ancillary material is used. Ancillary materials are manufactured under robust quality management system reduces the qualification burden of a CGT manufacturer. For this reason, it is preferred to source ancillary materials that are manufactured under CGMP. So a manufacturer will need to evaluate the performance in the manufacturing processes as well as the impact of lot-to-lot variability on the final products. Animal origin-free ancillary materials should always be the goal. It's desirable because it helps to reduce the adventurous agents, which is still one of the main regulatory fouling deficiency for CGT customers. Animal origin-free products are exempted from such viral testing. So with this in mind, most of our CTS branded products are designed to be animal origin-free and are well supported with regulatory documents. So ancillary materials can include animal, human-derived components, or viral vectors. Each carry with them specific risks that needs to be assessed and controlled when used in CGT manufacturing, as listed in the table here. There should be also demonstration to the authority on the level of control and testing to ensure the safety of the final CGT products. For starting material of human origin, this should be tested for human-specific viruses containing traceability data and traceability data from the transmission of human donor screening and eligibility information. In October 2023, the FDA also published the guidance for Industry Voluntary Consensus Standards Recognition Program for Regenerative Medicine Therapies. This is designed to identify and recognize voluntary consensus to facilitate the development and assessment of regenerative medicine therapeutic products. So the use of this recognized VCS can assist customers as well as manufacturers in meeting regulatory requirements more efficiently and increase regulatory predictability for CGT products. ISO 20399 is one of the regulatory standards for ancillary materials. It provides the general requirements around ancillary materials used in the further manufacturing of CGT products by ensuring the quality, consistency, and safety of such ancillary materials. So in a way, ancillary materials that comply to ISO 20399 can be inferred to be of a lower regulatory risk and better accepted during registration. So this is also the standard that our CTS media and reagents comply to. So ancillary materials per USP 1043 includes both the reagents and equipment components. A qualification program for ancillary material used in cell gene therapy should address the following areas: identification, selection, suitability for use in manufacturing, characterization, vendor qualification, and quality assurance and control. So by complying to the USP 1043 standards, also help to minimize the quality risk that arises from the use of substandard ancillary materials. So in this table, USP 1043 also emphasizes the importance of proper ancillary material selection and describes 4 tiers based on the classification of ancillary materials. So tier 1 includes the low-risk, highly qualified materials that are approved, such as the approved pharmaceutical products. So tier 2 are the low-risk, well-characterized ancillary materials, such as the GIPCO CTS products. So tier 3 are those materials with moderate risk and are not intended to be used for ancillary materials, which includes the IUO reagents. Tier 4 are the high-risk materials that do not comply with the cGMP standards and may also contain toxic chemicals or viral risk. So hardware and software should also be qualified and associated processes should be validated. Consumables are very important because they can ensure the safety of the product. As cellular products cannot be sterilized at the end of the production, they are produced under aseptic conditions. So equipment that utilizes the single-use consumables such as tubings, bags, and kits should be used. This will actually allow for processes to be performed in a lower grade clean room. So single-use consumables has direct contact to the CGT products, and it should be well characterized and having adequate validation data, such as biocompatibility, sterility, stability, extractable, functionality, and performance to meet the intended use. So when customers or manufacturers are using open processing steps, a suitable environment should be required. Successful incorporation of equipment into the manufacturing flow of cells for therapeutic use includes the maintenance of sterility and integrity, which is achieved by using suitable environmental controls, connectors, closed systems to maintain such consistency, as well as to exclude potential external contaminants. So in the following slides, I'll be moving on to the CTS portfolios. So the CTS branded products are GMP manufactured products designed for CGT to support customers' transitions from discovery through to clinical as well as commercialization. Our media and reagents undergone extensive QC testings such as mycoplasma, endotoxin, and sterility. The facilities that manufacture these products are also ISO 13485 certified, which is in accordance with medical standards. They also comply with US and EU guidance specific to ancillary materials, such as 21 CFR Part 820 and USP 1043. Comprehensive documentation package are available for customers such as the COA, COO, DMF, as well as RSF. So these products are also used in FDA as well as EMA approved CAR-T therapies and in more than 200 clinical trials. So our CTS cell therapy system instruments and equipment systems will include the raw tier system, the Xenon, as well as Dinoselect system. So similarly to our CTS media and reagents, this equipment system and consumables undergone extensive validation testing and comply with the regulatory guidance, include regulatory documentation package to support customers in their IND as well as NDA filing. So in the slide here, it actually features the type of regulatory support documents that are available to support the customers. So master file can be useful in regions that support master file processes for ancillary materials, and these include the US, Canada, and Japan. However, there are also certain regions that do not have master file processes for ancillary materials, and such information can be provided in the regulatory support file or the RSF. The RSF can be provided to customers or manufacturers under a CDA. So the quality of ancillary materials can affect the stability, safety, potency, and purity of a CGT product. To minimize such risk, it's necessary to implement ancillary material qualification program and to select ancillary materials that are well characterized and manufactured according to CGMP standards. The supplier should also have a documentation to ensure the traceability, consistency, suitability, purity, and safety of the ancillary materials. With the level of regulatory complexity, there's actually no one-size-fits-all approach. It's essential to connect with the suppliers and evaluators at an early stage to avoid unnecessary burden and cost. It's recommended to select CTS branded products over products that are RUO. So any change to the manufacturing should be adequately assessed and documented. So with this, I come to the end of the presentation, and I believe there will be Q&A from the audience as well.
Yes, thank you, Bingrong, for that informative presentation. We will now start the live Q&A portion of the webinar. Again, to our audience, if you have a question you'd like to ask, please do so now. Let's go ahead and get started. Our first question asks, does bioprocessing equipment systems for further cellular therapy manufacturing need to be registered with the health authority? And does the health authority require a medical device for this intended use?
Yeah, thank you so much, Casey. I think this is actually quite a common question that's been asked from the customers as well. So for bioprocessing equipment for further manufacturing of cell therapy, does not require registration with the authority. So it's recently also been mentioned in the consideration for the development of CAR-T products that the medical device classification is not required for bioprocessing equipment for CAR-T manufacturing. So this is the same understanding that we have from a global level. However, as the bioprocessing equipment can directly impact the CQA of the final product, the equipment system to be used, especially for those that are in direct contact with the cell therapy product, it should be validated, particularly the consumables, as well as the tubings. So from a risk perspective, the drug manufacturer should also qualify the supplier and select products that are well characterized and validated.
Wonderful, thank you. Our next question asks, why is closed system manufacturing equipment important, and what are the documents available for customers' assessment?
Okay, yes. So the closed system manufacturing equipment is actually been designed to minimize contamination risk and to ensure the safety and the quality of the final product. So for our single-use CTS consumables, they are tested for primary packaging integrities to ensure there's actually no leak, which can be a source of microbial ingression during the use by customers. So in general, our recommendation would be for customers to refer to our validation guide as well as the RSF for further information. So the use of the closed system manufacturing equipment can help customers in ensuring the quality as well as the safety of their end products and to mitigate any form of a cross-contamination risk.
Great. And it looks like we have time for one more question. That question asks, when do we request for RSF and DMF?
Yeah, so I think this is also another quite common questions, and it's actually this is actually a good platform as well to share with the audience. So the RSF as well as the LOA for DMF, this is actually provided only for regulatory filing needs. So we actually need the customer in for the IND as well as NDA application numbers for the DMF filing route. The DMF submission is not available for all countries, as I earlier has mentioned. So for countries that do not officially have this path, for example, China is a good example, it's also good to check with the regulators for the acceptance before applying through the web link for the DMF. So Thermo Fisher is able to send the DMF directly to the authority for review, which comes together with the customer's application. So for countries without the DMF, RSF, which is the regulatory support file, will be another source that provides adequate evidence to demonstrate the safety and the quality of our products. And this can be provided to the customers under a CDA to support their IND and NDA filing needs. Thank you.
Thank you, Bingrong. Questions we did not have time for during the presentation, including those asked during the on-demand period, will be answered by our speaker via the contact information provided at the time of registration. Our panel discussion is up next and will begin immediately. To enter this discussion, you will need to close this window, then click on the button titled Panel Discussion found in the auditorium. We'll see you shortly.